Transcription occurs in the nucleus where DNA is used as a template to make messenger RNA. During the transcription, the DNA in the gene is used as a template to make messenger RNA strand with the help of the RNA polymerase. During initiation, RNA polymerase binds promoter region, which controls the majority of gene expression. DNA double helix are unwinded and opened by binding. During elongation, the RNA polymerase slides along the template DNA strand to code them. As a complementary bases pair up, RNA polymerase links nucleotides to the other RNA molecule. At termination, once the RNA polymerase reaches terminator portion of the gene, transcript of the DNA is complete and RNA polymerase, messenger RNA, and DNA leave from each other. The messenger RNA include region of exons, which code for protein, and intron, which is non-coding section. Introns have to be removed by spliceosome to use messenger RNA in the translation. This process is called intron splicing. The messenger RNA removed intron leaves the nucleus through a nuclear pore and enters the cytoplasm to begin translation.
The process of translation occurs in the ribosome, in the cytoplasm, or in the cellular organelles. In an mRNA, the instructions for building a polypeptide are given by RNA nucleotide read in groups of 3. These groups of 3 are called codons. There are 61 codons for amino acid, and one codon, AUG, specifies the amino acid methionine and also acts as a start codon to signal the start of protein construction. Also, there are stop codons, which tell the cell when a polypeptide is complete. In translation, the codons of an mRNA are read in order by molecules called transfer RNAs. Each tRNA has an anticodon, which is a set of 3 nucleotides that binds to a matching mRNA codon through base pairing. The other end of the tRNA carries the amino acid that's specified by the codon. Translation is divided into 3 steps: initiation, elongation, and termination. In initiation, the ribosome gets together with the mRNA and the first tRNA, so translation can begin. In elongation, amino acids are brought to the ribosome by tRNAs and linked together to form a chain. tRNA starts out in the middle slot of the ribosome, called the P site. Next to it, a fresh codon is exposed in another slot, called the A site. The A site will be the landing site for the next tRNA, one whose anticodon is a perfect complementary match for the exposed codon. Once the matching tRNA has landed in the A site, it's time to form the peptide bond that connects one amino acid to another. This step transfers the methionine from the first tRNA onto the amino acid of the second tRNA in the A site. Finally, in termination, the finished polypeptide is released to go and do its job in the cell. Translation ends in termination. Termination happens when a stop codon in the mRNA enters the A site. Stop codons are recognized by proteins called release factors, which fit neatly into the P site. Release factors mess with the enzyme that normally forms peptide bonds, and they make it add a water molecule to the last amino acid of the chain. This reaction separates the chain from the tRNA, and the newly made protein is released. The new polypeptide will hold into a distinct 3D structure and join with other polypeptides to make a multi-part protein.
And now we're going to be going over how the CTFR protein works in the body. First thing first, how everything fits in with each other. In a diagram, you can see the cells as well as the mucus layer and the epithelial layers of cells right under it. As shown in the diagram, the mucus lines the inside of the organ, in this case the lung as an example. The apical layer is labeled on the side of the epithelial cell that touches the mucus. But how does everything function? Well, on the apical layer you have the phospholipid bilayer, which is a semipermeable membrane that encases the entirety of the cell. The CTFR protein serves as a part of the protein channel in which it lines the edge of it in order so that certain molecules can pass through, since the inner of the cell has such a high concentration compared to the outer of the cell, not only with the molecules such as chloride move from the inside of the cell to the outside, but then since this is happening, osmosis also occurs where water will move from the inside of the cell to the outside of the cell. This causes the mucus to become slimy and slippery, which is what is needed in order for it to move along in the body and not become clogged up, which is what happens in case of people who have cystic fibrosis.